Good afternoon, ladies and gentlemen, and welcome. Welcome to all of you coming in out of the Santa Claus Parade to participate in this afternoon's public lecture. I'm Janet Rossand. I'm President and Scientific Director of the Gairdner Foundation, and I'm honored to be here today to join you, all of you, for the Gairdner Foundation's public lecture series presented by TELUS Health. And we're very grateful to TELUS Health for partnering with us on our public lecture series, which brings the Gairdner laureates to locations across Canada to share their groundbreaking research with you in an accessible, easy to understand, and relevant manner. So what is the Gairdner Foundation, for those of you who don't know? The Gairdner Foundation was established in 1957 with the pioneering contribution of James Gairdner, an active Canadian philanthropist and businessman. And his vision at that time was to set aside funds to give prizes to recognize the greatest researchers around the world in the area of medical research that impacts on human health and well-being. And his vision really lives on today, 60 years later. The Gairdner Foundation has three main missions. The first is to celebrate and recognize excellence, and we do this through the Canada Gairdner Awards. Each year we present 7 awards. We just had a gala presentation for the awardees for biomedical and global health research. And over the years we've given out 377 awards. 87 of those awardees have gone on to receive the Nobel Prize. Our second mission, we don't just celebrate, we want to use the prestige of the awards to bring science to the next generation of students and the public. And every year, our 7 current laureates, along with past ones like Dr. Kandel, tour the country on an interactive lecture circuit, meeting with trainees, researchers, and industry leaders to share their exciting discoveries. And importantly, they speak about their careers to high school students to inspire the next generation of Canadian thinkers, entrepreneurs, and leaders. And of course, we host public lectures like today to engage the broader public about the excitement of science and its impact on our daily lives. So the Gairdner Foundation is very grateful to the support of the Government of Canada, the Government of Ontario, CIHR, for allowing us to put on important events like today. And if you enjoy today's event, we have another public lecture coming up on December the 4th, this time at the Ontario Science Centre, where Dr. David Julius, who's one of our 2017 laureates, will be discussing how our senses enable us to interpret and experience the world. And the title of his talk is From Peppermints to Pit Vipers. Intriguing, huh? So be there. As I noted, we're very grateful for the support of TELUS Health as the presenting sponsor of our public lecture series. And I'd like to invite Elaine Chin, Chief Wellness Officer at TELUS, to the stage to say a few words. Elaine.
Good afternoon. How many of you saw Santa Claus? Did you see Santa Claus? Oh, Dr. Kandel, she picked you over Santa Claus. Well, I'm honored to be here today supporting the Canada Gairdner Awards. I have to say to you that I'm quite the Gairdner junkie before I even joined TELUS Health. This is my 10th year supporting this organization, and I know a lot of you gasp at hearing that we are actually great pickers of the Nobel Prize winners. So I like to think of us as the Golden Globes before the Academy Awards. Yeah. So at the end of today, besides learning about Dr. Kandel and all the work, I would love each of you to be ambassadors of the Canada Gairdner Awards and tell everybody that this is a prize and a jewel in Canadian awards. It's a global award. So the foundation, as you know, is really focused on innovation and their commitment to building future thinkers. And so it's well aligned to our own values at TELUS, where we have the courage to innovate and a passion for growth. And more than a decade ago, we made a defining decision at TELUS to leverage our technology to tackle the #1 social challenge of our lifetime, as Mr. Entwistle will say, healthcare efficiency and effectiveness. So how many of you know that TELUS is in healthcare? Oh, okay. Since then, a decade ago, TELUS has invested more than $1.6 billion to bring patient-centered solutions to market and improve the flow of patient information across the continuum of care. And today, 1,700 TELUS health team members, including doctors, nurses, pharmacists, and other healthcare professionals, are delivering our strategy to be leader in healthcare ecosystem IT. So through our solutions, such as the electronic medical records that many of you may not know is at TELUS, from TELUS at the office, at your doctor's office, to home healthcare monitoring, to soon the ability for you to electronically transfer a prescription from your doctor's office to the pharmacist, is TELUS technology. So we are driving a shift from remediation to prevention of disease by empowering you, Canadians, to better manage the health through access to health information and technologies. So as an organization, TELUS is deeply committed to improving the lives of you, your Canadians, and our community. So we are proud to share this commitment with our dearest friends at the Gardner Foundation, and strongly believe that through our collective efforts, we will be able to create a friendly future for all citizens and deliver remarkable human health outcomes. So once again, on behalf of the entire TELUS health team, we welcome you to the Gardner Public Lectures, and we most certainly are humbled in having Dr. Kandel join us today. Thank you.
Thank you, Elaine, and thank you again to TELUS. So now it's my pleasure to introduce Dr. Kandel. So Dr. Eric Kandel is University Professor at Columbia University in New York, Kavli Professor and Director of the Kavli Institute for Brain Science, Co-Director of the Mortimer Zuckman Mind-Brain Behavior Institute, and a Howard Hughes Medical Institute investigator. And his research over many years has really pioneered our understanding of how memory works. And his research has had impact on age-related memory disorders, post-traumatic stress disorders, nicotine, alcohol, cocaine addiction, many areas of research about how the brain works and how things can go wrong. And because of his enormous contributions, he has a list of prizes and recognitions that are very, very long and would take me the whole lecture to give you. I will simply tell you that he's received 23 honorary degrees, and among the numerous awards that he's achieved is the 1987 Gardner International Award for elucidating the mechanisms of learning and memory in nerve cells, the Albert Lasker Award, and of course, the Nobel Prize for Physiology and Medicine in the year 2000. I've no doubt you're going to be inspired by Dr. Kandel, and I'll now ask him to come to the stage and share his research with you.
Thank you, Janet. After that introduction, I should probably best sit down. I'm really delighted to be here. I felt privileged to get the Gardner Award, and like everyone who gets the Gardner Award, which is the Nobel Prize of Canada, it certainly facilitates the evaluation of your candidacy by the Nobel Prize Committee. And I'm delighted to be here. I very rarely, oh, aren't you nice? Thank you. Thank you very much. I'm very rarely in Canada, and I'm really enjoying myself a great deal. So let me tell you a little bit about an overview of memory storage and end up with some recent work. Let me give you a little bit of background. As you probably know, the modern study of memory storage began where? In Canada, of course. And it began with the systems of problem of memory storage, which I'm going to turn to first. Where in the brain is memory stored? And I then go on to the molecular problem of memory storage. What are the mechanisms whereby storage occurs at each site? Let me begin with the systems problem. Where in the brain is memory stored? This began here in Canada, Montreal, unfortunately, not Toronto, but next time it'll be Toronto. He asked whether memories can be stored to specific regions of the brain. And he was a neurosurgeon who specialized in removing scar tissue from the surface of the cortex, which occurs following concussion. Brain injury that occurs when your skull bashes against the ground or something like that. And he realized that the brain has no pain receptors. So you could stimulate the surface of the cortex and see whether you elicit responses from the subject you're operating on, and make sure that when you're removing the damaged part of the cortex, you don't take with you important other areas of the brain. And in so doing, he really mapped lots of regions of the brain, and what he realized that when he stimulated the temporal lobe, he could elicit memory storage. Now, I was around when this was happening, and I can tell you the excitement that it caused. I was interested in psychoanalysis at that time, and Larry Kuby, one of the leading psychoanalysts in New York, and in fact in the United States, ran up here with a tape recorder to see what were the sorts of things that were coming out from these unconscious memories that patients were emitting as a result of these procedures. It created an enormous amount of excitement. And we learned even more about it when William Scoville, a neurosurgeon, removed not one, but both sides of the medial temporal lobe for somebody who had severe seizures as a result of having lesions in both sides. Penfield and Brenda Milner only made unilateral lesions. Scoville produced bilateral lesions because the guy, because the guy had bilateral lesions. And as a result of this, the patient, HM, never had seizures again. He was controlled with very low levels of medication, but he was left with a very profound amnesia. Brenda Milner, who was studying with Penfield and had been examining memory storage in the medial temporal lobe, went down and examined this patient, HM, as a result of William Scoville's request, and characterized HM's memory deficit. What she found was that HM had memories prior to the surgery. So he remembered his early experience in elementary school and high school, the early jobs that he had. So clearly those long-term memories were stored elsewhere in the brain. They were not stored in the hippocampus. He had perfectly good short-term memory. So if you asked him to remember a telephone number, 884-5447, he could play that back without any difficulty. So short-term memory is stored elsewhere. What was disturbed was the ability to convert short-term memory to long-term memory. So this provided the first evidence that a function of the hippocampus is to convert short-term to long-term memory, particularly new short-term to new long-term memories. And these were HM symptoms. He could not convert a new short-term memory to long-term memory, but he could learn new motor tasks. This was an amazing surprise that Brenda Miller discovered after working with him for 10 years. She found that if he did a mirror writing task, which is ask him to do the outlines of a star, without looking at his hand, without looking at the pencil, without looking at the paper or the star, looking into mirror, Leonardo da Vinci-like, he could do this as well as you and me. So the first day, he made a number of mistakes, but he got progressively better. Second day, he started already at a better level and was almost perfect. And the third day was perfect. When you asked him, HM, how come you did so much better on Wednesday than you did on Monday? He would say, what are you talking about? I've never done this before in my life. And this is an amazing thing, that people can learn tasks that are quite complex, but they involve different memory systems than the explicit memory that you use for conscious recall of information about people, places, and objects. Let me give you one example of this. Clive Waring had a severe bitemporal lobe lesion. As a result of it, he had a severe memory deficit. But nonetheless, he could do very complicated things.
How many years have I been ill?
About 20.
About 20. Can you imagine what to have one night 20 years long? With no dream? That's what it's been like. Just like death. No difference between day and night, no thoughts at all. In that sense, it's been totally painless. Which is not something which is very desirable, really, is it? Because it's precisely like death. If you have no senses of pain, you have no sense of any kind working either. I don't remember sitting down on this chair, for example, or the settee as it is. That was unknown to me. I've never seen a human being since I've been... That's the first photograph I've seen of anybody.
And who is that photograph?
It's one of my sons. I can't remember his name.
Tragic.
Clive Waring has one of the worst cases of amnesia in the world.
I know it's like to be dead now. Day and night the same, blank. No difference between dreams or anything like that, no senses at all. The brain has been totally inactive. No dreams and no thoughts of any kind whatever.
Clive was a renowned conductor living in London when he was struck down by a virus in 1985. Parts of his brain were completely destroyed, including his memory. However, his ability to play music is unaffected.
Do you feel different when you play music?
I've never heard a note since I've been ill. I don't know what it's like to play music when you're unconscious.
You played us some music about 2 minutes ago.
Not known to me. Totally unknown. I've never heard a note yet.
I think this is the most remarkable feature about memory storage. As I'll show you in a moment, there are two different kinds of memories. Explicit, which is a conscious recall of people, places, and objects, and learning how to play the piano is part of that. And implicit is motor and perceptual skills of various kinds. After a while, lots of things that are explicit, very hard to manage, become automatic, become implicit. So you can damage the hippocampus, which is absolutely required for acquiring this skill. But after you've mastered the skill, you don't need the hippocampus to recall it. And that's what you're seeing with him. It's just absolutely amazing. So clearly, that indicates there are two kinds of memories: implicit unconscious recall and explicit conscious recall. Implicit memories for motor and perceptual skills involves the amygdala, the cerebellum, or simple reflex pathways, while explicit memory storage for facts and events involves the medial temporal lobe. But one thing important to keep in mind is explicit memory, no matter how sophisticated, will after a while, as you master the skill, become implicit. Let me turn to the second problem, which is the molecular problem of memory. What are the molecular mechanisms whereby storage occurs for each of these forms? Now, it turns out that both implicit and explicit memory have certain features in common. Number one, there are stages in memory storage. In each case, there's a short-term memory that lasts minutes, a long-term memory that lasts days, weeks, or the lifetime of an individual. Number two, usually practice makes perfect. Repetition converts a short-term memory to a long-term memory. And three, long-term memory requires new protein synthesis as a result of alterations in gene expression. There are a number of experimental systems that have been used for studying implicit and explicit memory and drawing comparisons with them. I'm going to show you examples of implicit memory storage on the marine snail Implicit, which I've worked on a good part of my career. Here, an implicit memory does not require conscious attention. Implicit doesn't have much conscious attention. In contrast, explicit memory requires studies in mammals and is usually studied more recently in mice, where you can do genetic interferences of various kinds. And that requires conscious attention for memory acquisition. So in implicit, we've studied learned fear, memory for sensitization, and that involves alteration in a simple reflex pathway. In explicit memory studies, we've still focused on the hippocampus and looked at place learning and memory for space. Let me begin with implicit. As you can tell at a glance, this is a highly intelligent animal. This is exactly the sort of animal any one of you would select for the study of learning and memory. As a result of this, this animal has been highly recognized. Not only did it get the Gardner Award, it got the Nobel Prize. There are many people, including some in the audience, think that the animal rather than the investigator should have gotten the Nobel Prize. I must tell you, I showed this slide at the Nobel Prize lecture. Needless to say, the audience went wild. This is the human brain, your brain and mine, 86 billion neurons. Implicit has a much simpler brain, 20,000 neurons. Moreover, these 20,000 neurons are distributed in 10 ganglia, so each ganglion has only 2,000 nerve cells. Moreover, not only are there few cells, but each ganglion controls not one but several different behaviors, so the number of cells committed to a single behavior can be quite small, less than 100. Moreover, these cells are gigantic. Before I became presbyopic, I could see them with my naked eye. They are not only gigantic, but they're very distinctive in their location and their coloration. So you can see the same cell in the same position all the time and give it a name. Harry, Janet, Tom. We are more prosaic. We call them R2 if it's on the right side, L7 if it's on the left side. But the point is, these are uniquely identifiable cells. They're there in the same location with the same characteristics, unless you've taught the animal something and changed the characteristics of the cell. With this simple animal, with a simple nervous system, we focused on a very simple behavior. This is the head of the animal. This is the tail of the animal. It has an external respiratory organ called the gill, which is covered by a sheet of mantle shelf for protection, which has the residual shell that the animal has. And the mantle shelf ends in a fleshy spot called the siphon. If you now apply a weak tactile stimulus to the siphon, you get a brisk withdrawal of the gill. If you now do the same thing again, but first shock the animal's tail, so you scare it, learn fear, sensitization, then the same weak tactile stimulus that produced a modest withdrawal will produce a much more powerful withdrawal. And the duration of this effect varies as a function of number of noxious stimuli that you give. If you give one shock, you produce a short-term memory which lasts minutes. If you give 5 or more shocks, you produce a long-term memory that lasts days and even weeks. Let me show you the neural circuit. There are 24 sensory neurons that pick up from the siphon skin. They make direct connections to the motor neurons, and motor neurons make direct connections to the gill. If you stimulate the tail, you activate modulatory neurons. Modulatory neurons end on the sensory neurons, including on their presynaptic terminals. And there they act to alter the strength of synaptic connections. If you give one tail shock, you lose a short-term memory, which involves covalent modifications of pre-existing proteins and the alterations in pre-existing connections, and that's mediated by the activation of cyclic AMP and the cyclic AMP-dependent protein kinase, pKa. But if you give repeated tail shock, you activate gene expression, gives rise to the growth of new synaptic connections, and it's the growth of new synaptic connections mediated by the turning on of gene expression mediated by CREB, cyclic AMP response element binding protein, that carries the memory in the long term. The growth is really quite spectacular. Here's a sensory neuron, here's a motor neuron, in control animals. After long-term sensitization, you see a doubling in the number of synaptic connections. So let me just show you what this looks like. You stimulate the tail, you activate serotonergic modulatory neurons. They act in a 7-transmembrane receptor that activates an adenyl cyclase, increases the cyclic AMP-dependent protein kinase, which with a single shock causes an increase in calcium influx and increase in transmitter release from the presynaptic terminals. But if you give repeated tail shock, you increase the cyclic AMP level more profoundly. As a result, the cyclic AMP-dependent protein kinase translocates into the nucleus, carrying with it MAP kinase. MAP kinase removes the inhibitory constraint on transcription, which is mediated by CREB2, it's a repressor, and allows cyclic AMP to activate CREB1, and CREB1 acts on genes to give rise to the growth of new synaptic connections. And that's the staying power of long-term memory. Implicit memory can be studied in the snail, but you cannot study conscious memory in the snail, but you can study that in mice, which has a medial temporal lobe and hippocampus involved in memory for people, places, and objects. And hippocampus is extremely important for spatial memory. So for example, this is, I don't know very much about Toronto, but in New York, if you ask a cab driver to take you along Broadway from 100th Street to 110th Street, he says, what? How do I get there? But in London, you really have to pass tests to become a cab driver. You have to really know where you're going. And as a result, London cab drivers really know their stuff. If you ask them the route from Hyde Park to Primrose Hill, the hippocampus actually lights up. And the longer they drive a taxi, the larger the hippocampus becomes. And it is this system, which involves in spatial recognition and spatial memory, that is activated. The longer they drive a cab, the larger hippocampus becomes. And when they stop driving, the hippocampus regresses. And what's the take-home lesson from that? Don't retire. I'm trying to follow that model. Hippocampus is essential for explicit memory, spatial memory in mice. So that involves sight, touch, smell, position sense coming in association cortex, activating the entorhinal cortex, which contacts the hippocampus both directly and through the perforant pathway via the dentate gyrus. So these pathways get strengthened when an animal learns a spatial task. And how does that occur? The early part, very much like implicit memory, but a different second messenger system. So glutamate is released from the presynaptic neuron. It acts on an NMDA receptor, which activates calcium chemogene-dependent protein kinase, which phosphorylates the NMDA receptor and activates transiently synaptic transmission here. With repeated stimuli, it also recruits a modulatory system, and then it's very similar to the implicit system. Activates cyclic AMP-dependent protein kinase, CREB-1, downstream targets, and the growth of new synaptic connections. If you block PKA and prevent transcription, you dramatically interfere with long-term memory. So clearly, the subsequent steps are critical for the growth of new synaptic connections and for long-term memory. So both explicit and implicit memory storage use modulatory transmitter, and after the initial steps, they're remarkably similar. They both involve CREB-activated transcription and the growth of new synaptic connections. Let me turn to the memory disorder in the aging brain. But I just want to tell you one thing to reassure some of the young people in the audience. They see that long-term memory involves transcription and therefore produces gene alterations in the brain. And some young people in the audience may be nervous. They say to themselves, oh my God, gene expression in my brain? I was planning to go home tonight and make love to my partner. We're planning to have a baby. And that baby has to have engraved in the brain this stuff that I heard at this lecture. I'm sorry. Did you hear what I said, or shall I repeat it? I'm going to have to remember, that baby will have to remember all that drivel that I heard in the lecture today. It's a good thing I spoke more softly. I can reassure you that these gene alterations occur in selected neurons in the brain. They don't occur in the ovary. They don't occur in the sperm. You have my permission to do whatever you want tonight. Nothing untoward will happen to your brain. So both of them use modulatory transmitter and CREB-mediated transcription to turn on long-term memory storage. Let me now turn to the second topic, which I've become interested in recently, memory disorders in the aging brain. Not a moment too soon. Now, this is a recent phenomenon. Why has this become a recent phenomenon? The aging population. 1900, people didn't live long enough to have age-related memory loss. The average age was 50 years. Now, the weaker sex lives 76 years, and the stronger sex, 81 years. Amazing difference. Actually, just a remarkable difference. We never heard very much about Alzheimer's disease when I was in medical school in the 50s, because people under 70 practically never get it. It becomes common after 70. So 3% of the people age 70 have Alzheimer's disease. 90% of people, 50% of the people in their 90s have Alzheimer's disease. But as people lived longer, in addition to getting Alzheimer's disease, they seem to show a more modest kind of memory loss, and it wasn't clear whether this was an early stage of Alzheimer's disease or a separate sign of aging, just like the muscle mass ages. It is called euphemistically benign senescent forgetfulness, the memory loss with normal aging. And I've become interested in the problem. I've begun to explore it from a number of perspectives. The question is, are they distinct entities, or is benign senescent forgetfulness an early phase of Alzheimer's disease? To answer this question, my colleagues and I compared three features: age-related memory loss and Alzheimer's disease, age of onset and progression, anatomic localization, and molecular defects. Let me begin with age of onset and progression. If age-related memory loss is only an early phase of Alzheimer's disease, then if you go to a situation in which you never see Alzheimer's disease, you shouldn't see this age-related memory loss. And that's the case in mice. You go to mice, they have no Alzheimer's disease unless you put an Alzheimer's gene into them. Naturally, they never have Alzheimer's disease, but they have beautiful age-related memory loss. I'm not sure they think it's beautiful, but the investigator thinks it's beautiful. is an anatomical distinction between Alzheimer's disease and age-related memory loss. This is something investigated by my colleague Scott Small. People had known that Alzheimer's disease begins in the entorhinal cortex, and he had found that age-related memory loss begins in the dentate gyrus. We then carried out an experiment together in which we looked at molecular defects that vary as a function of age that specifically occur in the dentate gyrus and not in the entorhinal cortex. We used gene chips and we examined in postmortem material the dentate gyrus of people ranging in age from 38 to 90. We found a number of transcripts that changed systematically, and we're going to look at some other ones, but we began with one which, number one, changed in a very systematic way, only in the dentate gyrus, and we knew from before had an important physiological function, RBAB48. This simply just shows you, this is the dentate gyrus in the entorhinal cortex. The messenger RNA for RBAB48 changes systematically, as does the protein level. It doesn't change in other areas of the brain. What is RBAB48? Well, most of you know. It's part of the CREB switch that converts short-term to long-term memory. Activity like PKA phosphorylates CREB. CREB recruits the CREB binding protein, and the CREB binding protein recruits RBAB48 to alter chromatin structure and initiate transcription. So this allows for chromatin remodeling and gene expression. Humans allow correlations, but we can now look at causality by looking at mice. Is the level of RBAB48 also selectively decreased in the dentate gyrus of aged mice? Dentate gyrus, young and old, CA3, CA1, nothing. So you see exactly what you see in humans, and you see this now in mice. And now you can do some nice experiments. This is a young mouse, has perfectly good RBAB48, an old mouse, low levels of RBAB48. So an old mouse is very bad at this novel object recognition task. You put a mouse in the presence of two identical objects, it spends an equal amount of time with each. But if you now substitute a novel object for one of the familiar ones, it spends more time with the novel object. If you now inhibit RBAB48 in a young mouse, it will have a memory defect for this task. So even though it's a young mouse, if you inhibit RBAB48, it's enough to do this. If you have an old mouse and you increase the level of RBAB48, you rescue this memory deficit that normally occurs with aging. Does the inhibition of RB48 in mice also recapitulate the imaging pattern? There's a dysfunction in the dentate gyrus that you see with imaging. And Scott Small has studied this and shown that, again, inhibition of RB48 does exactly what aging does. So let me summarize. The clearest evidence so far that age-related memory loss and Alzheimer's disease are distinct diseases are that, one, age of onset of memory loss, age-related, is in the 40s. So at midlife, kids, so study now hard, because you're going to need all of that stuff later on. Alzheimer's disease much later. Anatomic localization, dentate gyrus, stays restricted there. Alzheimer's disease begins in the entorhinal cortex, but spreads. Molecular defects, humans show a linear decrease in RBAB48 messenger RNA protein. Old mice have decreased RBAB48. Young mice with reduced RBAB48 show memory defects identical to those with age-related memory loss. Alzheimer's disease is different from a molecular point of view. First of all, it begins in the entorhinal cortex and doesn't stay there. It spreads. There is no RBAB48 abnormalities and A-beta toxicity, as you all know. Does the aging body act on the aging brain? Are there hormonal factors that contribute to hippocampal dysfunction observed with cognitive aging? And might exercise help overcome these blood-borne insufficiencies? Now, you Canadians are very brisk outdoors. You ski and you walk. This might be of interest to you. So those of you who've been sleeping, maybe you wake up for a second. Turns out that there's a surprise here. It turns out that bone is an endocrine gland, and it releases a hormone called osteocalcin. Osteocalcin acts on the testes, on the pancreas, on muscle, on the liver, but it also acts on the brain. When it acts on the brain, it gets through the blood-brain barrier, it prevents anxiety and depression, and favors spatial learning and memory. So we began to study this to see whether it affects age-related memory loss, We saw that direct administration of osteocalcin into the dentate gyrus, which is critically involved in age-related memory loss, enhances contextual field conditioning both in young animals and in old animals, so it overcomes age-related memory loss. But only if you inject it in dentate gyrus, which is critical for age-related memory loss, has no effect in CA3. Osteocalcin cannot ameliorate age-related memory loss when the function of RBAB48 is inhibited in the dentate gyrus. So clearly, osteocalcin acts through RBAB48. Behavioral effects of osteocalcin administration is a function of age. Aged mice show a decline in memory performance. This is a young mouse. This is an aged mouse. This is a novel object recognition task. But if you give an aged mouse osteocalcin, it reverses this age-related memory loss. If it's administered directly into the dentate gyrus, if you inject it elsewhere, there's no effect. Osteocalcin administration also enhances the memory performance of young mice. This is aged. Aged osteocalcin shows it overcomes age-related memory loss, but even a young control, osteocalcin is good for you under all circumstances. Osteocalcin injected directly into the dentate gyrus increases levels of PKA, CREB, and RBAB48. These are all components. PKA activates CREB, and CREB recruits RBAB48. So, whoops. All of these components of the switch that convert short-term to long-term memory are enhanced when you inject osteocalcin directly into the dentate gyrus. Conversely, osteocalcin knockout mice have low amounts of CREB and RB48. Here's a knockout, low CREB, low RBAB48. Systemic osteocalcin passes through the blood-brain barrier and accumulates in the dentate gyrus in CA3. And you can see that quite clearly here, where you can recognize when an antibody reacts with both endogenous and recombinant osteocalcin. You can see clearly that it's there in dentate gyrus and CA3. Moreover, we've recently succeeded in recognizing a receptor, 7-transmembrane spanning receptor, GPR158, that is specific for osteocalcin. It binds osteocalcin very effectively. And knockdown of the receptor, GPR158, leads to reduction in the level of RBAB48 protein. So this is the control, the receptor, osteocalcin, RBAB48. If you knock it down, you see you have a significant decrease in GPR158. Knockdown of the osteocalcin receptor causes a memory deficit that, because you now have knocked down the receptor, is no longer rescued by osteocalcin. So this is novel object recognition. Even if you inject osteocalcin, you can't rescue it because you don't have the receptor. The circulating osteocalcin decreases throughout the lifespan. So as you age, one of the dramatic things that happens is that the bones release less osteocalcin. And the best way to overcome that is to do exercise that involves the movement of your bones. Walking, running is absolutely essential as you age. This is mice with osteocalcin, aged mice. They run around with saline, they do nothing. So I swim every day. That's a wonderful exercise. It's a very good substitute for psychotherapy. It doesn't do a goddamn thing to you in terms of bone mass. What you need for bone mass is to walk. 2 miles a day is what I would recommend to you as you mature. If you want to do more, you have my permission to do as much as you want. So let me summarize. Humoral and hormonal factors affect cognitive aging. Osteocalcin released by bone ameliorates age-related memory loss through CREB1 and RBAB48. Because aging is associated with decreasing bone mass, this decrease in osteocalcin could contribute to age-related memory loss. Conversely, this might explain the beneficial effect on cognition in the age of vigorous exercise, which builds bone mass. The reversibility of age-related memory loss strengthens the idea that age-related memory loss and Alzheimer's disease are distinct diseases. Exercise will not help you very much with Alzheimer's disease. A sound body assures a sound mind. So let me summarize. Age-related memory loss and Alzheimer's disease. Age-related memory loss begins early and progresses linearly. Alzheimer's begins later and progresses explosively. Anatomy, restricted to the dentate gyrus. Alzheimer's begins in the entorhinal cortex and spreads. Molecular defects, decreasing RBAB48 and cyclic AMP signaling, no cell loss or very minor cell loss. while Alzheimer's disease, entorhinal cortex, A-beta toxicity, a protein folding disorder, and major loss of nerve cells. So treatment for age-related memory loss prevents the onset of progression of the disease. Regular physical exercise to increase osteocalcin. Walking, very good to do even in Toronto. Good diet, low in animal fat, control of high blood pressure and diabetes. Cognitive challenges, take on a new task. Learn French if you don't speak it. Social involvement, continue to work and to learn and enjoy your lives. These are my colleagues with whom I'm privileged to collaborate, and I've been supported by the Howard Hughes Medical Institute, and some of these experiments have been carried out together with Scott Smoller's laboratory and Gerard Krinsecki. Thank you very much.
So now you have your instructions as how to behave and how to avoid age-related memory loss.
You're doing it you're too young to worry about it, but take my word for it.
I was going to say,
Keep on introducing speakers.
You seem to be doing very well, I have to say. So at this point, we'll open the floor to questions for Dr. Kandel.
Yes. Wait until the microphone comes around.
Yeah.
Oh, thank you. Hello? Hello? So you said that there's a progressive decrease in RBAB48 in the dentate gyrus, but that you can still rescue memory by injecting the osteocalcin in the dentate gyrus. So I'm wondering if there's a sort of timeline where you don't have enough RBAB48 for the osteocalcin.
Doesn't work anymore.
It stops?
Yeah.
Oh, okay. Makes sense.
First of all, wonderful talk.
Thank you.
Second of all, my understanding, not being directly in that field, is that new neurons are generated throughout life and migrate to the dentate gyrus.
They're generated within the dentate gyrus.
Okay, so how does that function in terms of the scheme?
It decreases with age, and when age-related memory loss really sets in, it sort of stops. I don't think, this is a controversial area in the field, I don't think it contributes very heavily to a reservoir of memory storage. I think it's a minor contributor, but it's a contentious issue.
I have two short questions. First of all, what used to be called presenile dementia, how does that fit into the age-related versus Alzheimer's? And secondly...
I didn't understand the first question.
When I was a...
Presenile dementia.
Yeah, is that age-related or is that...
Presenile dementia is a private terminology. I presume it refers to not age-related memory loss, but Alzheimer's disease. But one doesn't know. In the early days, one didn't know how to distinguish between age-related memory loss and Alzheimer's disease. One should re-examine those patients who have been given that diagnosis. Or simply with imaging experiments, you would pick this up. You have really destructive changes in the brain with Alzheimer's disease, while you don't have serious brain damage with age-related memory loss.
The second question is, I am a pediatrician, and I'm interested in what effects on memory, long-term memory, has child deprivation, both nutritional and psychological or physical.
Did you understand that question?
Yes, so he's a pediatrician, so in terms of childhood deprivation, either physical or mental, what's the long-term effect on?
Devastating. Have you watched some of the programs that Charlie Rose and I have done? We've done several programs on children who have been brought up in orphanages or other kinds of situations in which they've had serious deprivation. And it's absolutely catastrophic. So one is a very famous comparison that actually was done by a psychoanalyst. They very rarely do experiments in the 1950s, in which he compared kids brought up in an orphanage, in which one person took care of 8 children, and those that were brought up in a special nursing attachment to a woman's prison, in which the women prisoners were able to see their child 3 times a day and take care of it. And the children raised in a prison did just as well as kids brought up in Ontario, while those that were, brought up in these nursing homes with one person available for 7 or 8 kids did terribly. So attention to the child early is absolutely critical. All kinds of situations, even poverty, has bad effects on cognitive development of kids. Charlie Rose and I did a program on this. It's really quite striking.
I wanted to know your comments on a condition called transient global amnesia, where for a short period of time you stop recording explicit experiences. So you're okay, and then for like 3-4 hours you don't record anything that's happening to you. Nothing from a certain point on. It's like anterograde amnesia, and then it comes back later. Any idea what the mechanism is?
Did you get that?
Situation where you have a short-term memory loss, no ability to accumulate memories, and then it comes back in a few hours. So it's sort of transient.
I would have to guess. I don't really know what's going on. But one possibility is that, for example, the memory has been put in the hippocampus, which is sort of an intermediate storage site, and the hippocampus may have been ischemic, but only transiently so. So the information is there, and once the hippocampus recovers, you can have access to it. So it's a mild, reversible damage to the hippocampus, I would guess.
I'm so honored to have heard your talk. I have a question about GPR158, which is the osteocalcin receptor. I wonder if that gene expression of this receptor changes with age as well.
Does the expression of the receptor for osteocalcin change with age?
We have not looked at that. We're just starting to play with the receptor. That's a very good question.
Hi. My question, I'm very interested in your comments about hormones in relation to memory loss, and women of a certain vintage will sometimes refer to mental pause. And I'm wondering what we understand about hormonal changes in women and how that affects memory, different from the age-related memory loss that you described as linear.
My guess is there's a fair amount known. I happen not to know it. I'm serious. It's not an area I've really familiarized myself with.
Hello. I was wondering about the systemic osteocalcin. So osteocalcin is systemic, right? So why is it that when you inject it elsewhere, it does not have an effect or accumulate in the dentate gyrus?
I'm not sure I caught that. Do you want to try that again?
Try that again.
I think he was just asking why, when the osteocalcin is injected in other areas, if it is a systemic product, why it's not effective.
So, why does osteocalcin only work if you inject it in the dentate gyrus?
Oh, because that's where the receptor for osteocalcin is. It's specific to the dentate gyrus, and that's the area that is involved in age-related memory loss.
While she's getting there, osteocalcin is an amazing
Yes.
amazing molecule. So how do you think it has different receptors in different
receptors
organs?
in different tissues. Yes.
Interesting.
Yes. It's really a marvelous story to realize that bone is an endocrine gland that acts on many organs of the body, and recently its importance for the brain has emerged in a very powerful fashion. Gerard Kosenki is very important for that. All my work has been done in collaboration with him.
Thank you so much for your talk. Forgive me if you're not familiar with this body of research, but I study motor learning, and I'm interested in how exercise can impact motor learning. And there's research showing that when people exercise at a high intensity, immediately after they do a motor learning task, this enhances consolidation and later retention of implicit motor learning. But we know that chronic fitness, or not acute bouts, actually enhances verbal and cognitive reasoning better, but it seems that there's no effect on this acute bout where it happens right afterwards. Can you comment on what what your first thoughts are on the possible mechanism differentiating between the motor learning and the verbal learning, the effects?
I'm glad you're here, Janet. I wouldn't survive without you.
I will try. So, and she can correct me, there is a difference between acute exercise, which will enhance motor learning, whereas chronic exercise enhances cognitive behavior. Yes? So do we have any idea about the difference between acute and chronic exercise?
I think the issue is less acute chronic versus what kind of exercise you do. You can do swimming acute and chronic. It's not going to help your bone mass very much. It's not going to increase the level of osteocalcin. So if you're worried about this component of age-related memory loss, acute and chronic doesn't matter in that sense. But it is important in terms of walking. It's important to walk a lot. So the general idea right now is 2 miles a day would be a good, reasonable goal for people my age to undertake, minimally, ideally more. Fortunately, to walk from my hotel to here, I did more than half of my requirement for the day alone.
Dr. Kandel, this is a question about your personal history.
I can't see the person. Not that I will understand the question anyway, but I'd like to see the person.
Oh, okay, sure.
Oh, very intimidating, yes.
You mentioned how you studied psychoanalysis. What brought you initially to psychoanalysis, and is any of your work in molecular biology support or refute Freud's notion of the unconscious?
That's a good one.
What did he ask?
Oh, it's
Freud,
a good one.
yes.
So I will repeat it. So he said that you began with psychoanalysis, and as you've moved on to the molecular studies, have your molecular studies led you in any way to reconsider Freud's analysis of the unconscious?
Well, my work has not really related itself to analyzing unconscious mental processes, but I can say, having now maturity and distance from Freud and from Vienna, where I was born, how do I feel about Freud? I have enormous respect for Freud. I think many people don't realize his life story. Freud began as a neuroscientist, and independently of Cajal, he developed the idea that the neuron is the elementary unit of the brain. He developed the neuron doctrine. He had a classic paper on aphasia that some of the pediatricians in the audience may be familiar with. So he was really quite gifted, and he wanted to go on being an experimental scientist, which he was really first class. But in those days in Vienna, to do experimental science, you had to have a private income. The university didn't pay very much. I must tell you, things haven't changed all that much, but so he went into private practice. And when he developed the idea of unconscious mental processes, the nature of neurosis per se, he knew he was developing a theoretical scheme for which there was no biological support. He tried in 1897 to write a neurobiological paper. It's called Psychoanalysis for the Neurologist. There is a gentleman in the audience nodding his head. Try to read it. It's practically incomprehensible. He tried to draw a neural circuit for repression. It's hilarious. And he came to the conclusion after that we're nowhere near having the brain biology to explain any of the phenomena that he's doing. So he's developing an abstract scheme that has no relationship to neuroscience because the neuroscience isn't ripe to do it. But someday, neuroscience is going to come along and disprove a lot of the ideas that I'm developing because they're not based on any obvious biological truths. When there were possible biological truth, he tried to, develop the argument, but he was aware that a lot of stuff would be proven wrong. I think what's amazing is that a lot of this stuff is reasonably right. I mean, we do have unconscious mental processes. They vary in degree, blah, 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 blah. Dreams have some significance. So, I mean, he was wrong in a number of things, but general outlines, I think he did very well. But you got to remember, he was working, 1900 or a little bit earlier to when did he die, 1939 or something like that, 1940. Way behind the development of modern neuroscience. What is a shame is that current psychoanalysis is not more strongly biological based. The only person really doing it seriously is a guy called Mark Solms, and you'll have to ask me in private my opinion of that work.
Dr. Kandel, I'd like to hear your viewpoint as to the future cross-intersection between computer science and their willingness to want to go to implant instruments of you know, that will cross-sect with all the problems that we're talking about here today, and get around all that. And so what do you see in the future in terms of computer science of the human body?
Absolutely. I think one of the great futures for people interested in neuroscience, young people, is to bridge between computer science and brain science. So that's important for two reasons. One is, when I was young, there were people developing theories about brain function, of what kind of circuit was required to mediate this kind of body. It was all theoretical. It was nonsense. No one went back to examine whether or not these ideas were right, because we didn't have the tool to examine these things. Now, computational science is fact-based. People develop, like Larry Abbott at Columbia, is one of our colleagues, is spectacular in this area. There's no bullshit. He develops an idea, he tests it. If it's wrong, he drops the idea. And this, it's really changed. There's a much tighter coupling between theory and experiments. So that's very important. And computer science is very important to that, to testing some of these ideas, but also it's going to be very important, it is becoming very important, to developing control devices that allow patients that have been injured in some way with a stroke to be able to maneuver their arms or an object that serves as a substitute for their arms so they can feed themselves, et cetera, et cetera. So the interplay between computational science and brain science has a great, great future. It's just at the beginning.
First of all, it's great that you're here. Second, I want to mention the good news that Brenda Milner just turned 99. And that must tell
she
us
is unbelievable. She's my, why is she? Has she got a Gardner Award? She got a Gardner Award because that's a very intelligent committee. She has never gotten the Nobel Prize, and I simply can't understand it.
Yeah.
She is phenomenal.
She is indeed. I think she still has graduate students. The question I was asked, just realizing that there may be many in the audience who have family members or friends who have either Alzheimer's disease in early or late stages, and they read all the time about various clinical trials that are taking place. How would you summarize your take on the science and the prospects that's taking place?
I think the frustration of the pharmaceutical industry with the development of drugs against Alzheimer's disease is great. There is reason to believe that a number of the drugs that have been developed work. But the reason they're not effective in patients is by the time a patient has developed symptoms, they've lost so many nerve cells, which they can't restore, that you need to get to them earlier. So the same drugs that are available now, if one could give them to patients 10 years earlier, would have an impact. So the attempt right now is somehow either get people with a family history who are willing to start a drug, when they're 40, and do a large-scale trial, control group, an experimental group, things like that.
Hi. I'm wondering if low levels of calcium have any relation to I'm blanking, osteocalcin, and if abnormal levels of calcium could potentially lead to age-related memory loss.
Let me see if I got it right. So you're asking whether low calcium levels are related to osteocalcin and bone loss, et cetera, So as we age, we lose bone mass and calcium levels drop. Is that related somehow to production of osteocalcin?
That's a very good question, to which there probably is an answer to, but I don't have it. I don't know.
Gerard Casenti would probably know.
He would know immediately, yeah.
Thank you so much for your talk. Really appreciate it. It's really cool. I was just wondering if you have any pieces of advice for aspiring neuroscientists in the audience.
Advice for aspiring neuroscientists, of which there are many in the audience. What would you tell them?
You're privileged. It's a wonderful area. It's really expanding in a brilliant way. And I think you have a great career ahead of you. I think the challenge is to take the increasing amount of knowledge that we've gained from experimental animals and begin to apply this to human beings. to really improve imaging methodologies, which are, in principle, are powerful already, will become even more powerful, so we can study the human brain with the facility now we have, for example, with the mouse brain. I think the future is extraordinary, and the future particularly in human beings. Just to give you an example, we have a reasonably good understanding of a significant number of neurological diseases. Our understanding of psychiatric diseases is embarrassing. And to really put psychiatry on a rigorous biological basis is the challenge of your generation. There's no reason why this cannot be accomplished. And it's extremely important because if you see the number of people that have, serious psychiatric problems, it's terrible. So I think trying to bring the power of brain science to bear on psychiatric illnesses is a major challenge.
I have a question about Alzheimer's. I was watching a summit on the internet about all these natural ways that people can reverse Alzheimer's. And I don't know if you're familiar with that. They were talking about coconut oil, various herbs like radiolea, and many other things. And I wonder if you have any thoughts about that.
So the question is about various natural remedies that are proposed to prevent and delay Alzheimer's. Do you have any comments?
There are so many natural remedies out there, from everything from ingrown toenails to Alzheimer's dementia. Most of them are unreliable. One has to do outcome studies in a controlled way in order to show that. Some of these may very well be effective. At the moment, there's nothing out there that's proven. It's been established in a reliable way. No one has sold the FDA, for example, on the fact that something is out there.
Thank you.
I'm just going to bring a quick one. We have a bunch of people who are joining us on webcast as well. So we have a question from them. This is from James Kennedy, who's watching, and it says, roughly speaking, how much do you think human memory is genetic versus enhanced by training?
50-50.
Fair enough.
There certainly is a genetic component to it. You see this in families. But you also see people who really train themselves at this and get very, very good with this, whose parents don't have such spectacular memories. So I think both is involved. Certainly it helps to have genetic predispositions, but those of us who don't have those genetic predispositions, if we work hard, there's no reason why memory can't improve.
Thank you. I'm just wanting to, if you can discuss a little bit about the issue in post-traumatic stress disorder of repressed or memories that seem to be locked away that can be recovered, and their tendency to recur at trigger point times.
Did you get that one?
No.
Okay. So she's asking about in post-traumatic stress disorder, indeed in other situations where you can have locked away memories that can be reawakened by trigger points. Do you have any understanding of how that occurs?
Actually, we're working on post-traumatic stress disorder. It's very interesting because it's a very complex kind of disorder in which a traumatic event will set up a persistent change in the brain, so it can be elicited in a variety of circumstances. There's a large neural circuit that gets involved in that is shut down under many circumstances, but a variety of subtle clues that either physically resemble the situation that triggered it or somehow are associated with it in a way that the subject sees this as being a symbol, unconsciously sees this, that will trigger this. And it's a very fascinating disorder, which people are really beginning to get a handle on. There's sort of functional prionic mechanisms that are involved in that keep that memory going for a long period of time. And it's extremely important. And one of the things we have learned is early intervention is very, very important. If you have psychiatrists at the battlefront that will handle people as soon as they're exposed, if this is a battle-produced thing, then to initiate various kinds of support therapy and also desensitization treatment, which is very important. Having the person experience the same frightening thing but in a protected environment ultimately will cause habituation of the response to that. So there are many not many. There's several useful therapeutic devices that one can use, but one should get access to the person who suffers from it as soon as possible.
Yes. It could be maybe science fiction, but is there any artificial or biological network can be interfaced with the brain for memory storage or retrieval soon?
I didn't get that. Can you repeat it?
He was just asking whether there's any mechanistic or computer-based methods right now that can be interfaced with the brain, whether that's something that's present or whether it's something that's going to be coming in the future. So connected, I think, to the tech one.
Computers talking with the brain directly, is that now or is that coming in the future?
And specifically for memory.
I hope never.
For memory.
I think computers should be used as devices. They should not be substitutes for people. There's something about the social interaction of human contact that's very magical and special. I would not want to lose that in any way.
Okay, how are we doing? Yep.
So I'm wondering about like how you feel towards, for example, genetic enhancement of those receptors that you just talked about. Is that like anything that could be potentially used as any therapeutic kind of means in the future, since we have such powerful genetic engineering
What you're trying to get at is, you told us that with osteocalcin, not only can you reverse age-related memory loss, but actually young mice do better when you give them osteocalcin. So what are the genetic or therapeutic implications of that?
Young people should do a lot of walking. I'm serious. I think it's absolutely true. It's changed my thinking because, you know, I love swimming because it's so relaxing and because there's a nice pool right near me, and don't tell anybody, but I tip the lifeguards so they keep the temperature above 80. And if somebody complains, I call them an anti-Semite.
This is a rather broad question. I was just wondering your opinion on nutrition on brain health. For instance, the incidence of Alzheimer's in vegetarian societies.
Sarah, can you help? Nutrition?
Sorry, was a question about the impact of nutrition on brain health? So she was referencing cases of Alzheimer's disease within vegetarian societies and other.
Vegetarian? Effects of nutrition on brain health. Do we have any specific information relating to diet and early onset memory loss?
I'm afraid I really don't know enough about that. My guess is there is information available, but it's really not my specialty. I can tell you that, you know, for a variety of reasons, I very, very rarely eat meat unless I'm in a social situation, which would be awkward to, you know, get an alternative. And I have a very reasonable and healthy diet. Yogurt and fruit is my lunch. So I think having a good diet is very important, and not having too much meat, having more fish and dairy is very beneficial.
Can I ask another a related question, which is obviously one of the other events that occur in the brain that's related to memory loss are vascular events. And you mentioned on your lifestyle having no diabetes, low blood pressure, etc., etc. So there is, so that would be a connection.
Absolutely, absolutely. Particularly, with many of these things like high blood pressure, there are many ways of controlling that, with drugs and exercise in addition. But certainly with drugs, there's no reason why blood pressure should not be adequately controlled. You need several things with that. One is a clinician who takes it seriously with you, number one. And number two, those people who do have hypertension or a tendency to hypertension, to take their own blood pressure at home so they see how they're doing in a relaxed circumstance. There's something called a white coat phenomenon. Many people show a slight to moderate increase in blood pressure in the presence of a physician or a situation that scares them. But if they take it at home and they find that 95% of the time they're doing great, it's very reassuring to the physician and to the patient.
Okay, we're just going to have another quick one from everyone who's joining us online. This is from Melanie Martin, and it's, do hippocampal cells have an ability to communicate the concept of time to one another? And could the deterioration of this process be a factor in memory-related diseases?
So do hippocampal cells have the ability to talk to each other and measure time? So is there a time component to memory acquisition, and does this decay with age?
There are certainly memory components, time components to memory. They have not been very effectively studied, and how time fits into hippocampal function, which it does, is not very well understood at this point. Lots of stuff still to be explored.
Okay, where's the microphone?
Okay, you mentioned at the beginning that concussions result in scarring on the cortex. How do you think that impacts or maybe accelerates age-related memory loss?
What was the first part of the question?
It's the impact of scarring resulting from concussions on memory loss and the development of memory.
What's the impact of concussions and
Very
memory
bad.
loss?
I'm serious. Again, Charlie and I did a program on this. It's particularly important for young kids. You know, parents go out, they think it's great, their kids play football, they play soccer, they head the ball in soccer, they, beat each other up in football. Those things cause concussions. And concussions are bad for anyone, as we're beginning to learn from professional boxers whose brain is really beaten out as a result of their experience in the ring. Football players, many of them suffer from brain disorder as a result of this. Thank you very much. You're very nice. We should travel together from now on.
Hello. First of all, very honored to be here today. I just wanted to ask you, what's your advice for young scientists?
Your general advice for a young scientist?
I think you have to fall in love with the field that you're going to work in. So selecting a partner that you like and selecting a job that you like are two of the most important decisions you have to make. After that, everything else falls into place. I'm serious. I should tell you from my own experience, my partner is absolutely critical to many of the good things that have happened to me. I'll give you just one example. We were starting to date when I was in medical school. And I wanted to be a psychoanalyst. But in my senior year, I took an elective in brain science. And to my astonishment, I'd never worked in a lab. You know, reading a book is one thing, even principles of neuroscience. It's nothing as exciting as working in a lab, doing experiments with your own hands, seeing results, discussing, gossiping, et cetera, et cetera. And I remember telling Denise one night, I really am enjoying this stuff. I've never had an experience like this. I could see doing this for the rest of my life, but it's unrealistic. You don't have any money, I don't have any money, we want to get married and have kids. And she said, money's of no significance whatsoever. In the subsequent 61 years of marriage, she's not repeated that very often, but at that time, it made a tremendous impact on me. And when I finished my residence in psychiatry, I'd earlier gone to the NIH, and I'd done some very nice work there. So since most psychiatrists can read and write, somebody who gets out of a residency and has done some science is eligible for an advanced position. So I was offered a chairmanship at Harvard Medical School. Well, at Beth Israel Hospital. I would have killed for that 10 years later. But Denise said, what? Throw your job away for a lousy administrative job? It's crazy. And several points in which, my ego or something like that would have deviated because of money or resources or recognition. She really had the good sense to push me in the right direction. I would never have gotten the Gardner Award if it wasn't for that. I mean that, the kind of recognition that the Gardner carries with it.
We're going to take two more questions.
Hi, this is less of a question, though it's an honor to be here and to see your talk. But just that there was a couple of questions about the effect of nutrition and supplements on brain health. And the Alzheimer's Drug Discovery Foundation recently launched a web page called Cognitive Vitality, where they rank all the different supplements and whether they're of any value. So if people that are interested can maybe just Google that, Cognitive Vitality, the Alzheimer's Drug Discovery Foundation.
Comment, not a question. Okay. One more question. Gentleman over there. And then we can mingle.
Or get out of a wet clothes and into a dry martini.
A bit early for the dry martini.
I'd like to ask about the other aspect of dementia. We talked about memory loss, but I'm very curious about your insight in the impairment of cognitive reasoning, whether they are related or not related. So let me repeat that. In addition to memory loss, what causes the impairment or the impairment of cognitive reasoning? Maybe it's related, being not neuroscience. Maybe it's a wrong question. Cognitive reasoning, in addition to memory loss.
So I think cognitive reasoning and memory are two separate issues, although obviously they overlap. If you have no memory, all the cognitive reasoning in the world is not going to do much for you. But it's a separate trait. There's some people who don't have a spectacular memory, but they're extremely insightful in being able to figure things out. That's probably even more important than memory in some ways.
All right, I think we are going to...
Well, thank you very much.
Oh, you were great too.
Let's all meet over a glass of wine.
Yeah. Well, I think it's a cup of coffee at the moment. So I don't think I need to say how much everybody enjoyed it, because you've all just expressed yourself. Thank you so much for your lecture, your insights, and the little messages for our next generation of neuroscientists. So please, everybody join us at the back.